BRUIN trial is the phase 1/2 trial of LOXO-305 for patients with CLL and other B-cell malignancies. This is a first-in-human trial looking at the non-covalent BTK inhibitor LOXO-305, a dose-finding study where doses were ranging between 25 and 300 milligrams. This is an ultra-heavily pretreated patient population, particularly the CLL patients, all of whom, or the majority of whom, have had prior therapies like BTK inhibitors, chemoimmunotherapy, anti-CD20 antibodies, and the list goes on and on. These are, the median prior therapies for a patient who had a BTK inhibitor previously was 4. So a patient population in need of new therapies, a patient population enriched for progression of disease on BTK inhibitors, and therefore a unique patient population that hasn't been studied prospectively, and certainly not studied with an agent like LOXO-305, which is a completely unique BTK inhibitor in terms of the way that it works, different than the covalent BTK inhibitors. The major finding from this trial were twofold. Number one, LOXO-305 was incredibly well tolerated. When you look at the AE table that I present, there's only 3 AEs in total that cross the 10% mark, which is remarkable considering this patient population, but this would be remarkable for any CLL trial. And then some of the classic BTK inhibitor-associated adverse events like AFib, for example, which can be as high as 10 to 15% in other studies, at least at this particular time point, was less than 1%. Grade 3 hemorrhage was less than 1%. So safety was certainly achieved. No MTD was identified. No DLTs were observed, and the go-forward dose was determined to be 200 milligrams once daily. The other major finding was the activity. Again, in a patient population as heavily pretreated as this, we report response rates of over 60%. But even more importantly, that's with relatively short follow-up for the entire cohort. If you start to look across time, patients who are followed for 6 months or more, 7 months or more, 10 months or more, you start to see response rates that exceed 80%. And so this type of molecule doesn't come along that often in CLL, potentially represents a paradigm shift. And of course, nothing I reported today was a randomized trial. There was no comparator. And so this does open the door to future trials where this molecule will challenge the standards of care from today's times and potentially try to change the way that we manage patients with CLL, not only in the relapse refractory setting, not only in patients who have identifiable resistance mechanisms, but in general, this may be a very important class of molecules across the board for CLL.